In this module, we'll talk about Golgi apparatus and the lysosome. Golgi apparatus receives vesicles from the, from the endoplasmic reticulum. Endoplasmic reticulum packages proteins and sends to Golgi apparatus. Golgi apparatus is the site where the proteins will be sorted, they'll be further chemically modified, they'll be concentrated, enriched in a specific vesicle, and the vesicle will be sent to its respective destination. We have already talked that proteins have carbohydrate molecules attached to them which serve as the addressing system. These proteins are packaged and sent to Golgi apparatus, where further sorting and concentration of these proteins is increased in a vesicle and sent to the appropriate destination. Golgi apparatus, it looks like a stack of rotis. These rotis or these stacks, there are three different types and they have distinct functions. The one closer to the nucleus is called the cis cisternae of the Golgi apparatus. The ones in the middle are called the medial cisternae, and the one which are facing the plasma membrane or the cell membrane are called the trans cisternae. Vesicles are actively moved between these three different types of cisternae, and these cisternae are functionally distinct. They have different type of enzymes which are performing different types of reactions on these proteins. Golgi apparatus is the site where secreted proteins are enriched and sent to the lysosome. That's another organelle which we'll talk about next. And proteins are sent to the plasma membrane. Some proteins are also sent to the lysosome. That is the organelle we'll talk about next. Lysosomes are basically the organelles that get rid of the garbage of the cell. Lysosomes contain hydrolytic enzymes. These are the enzymes that can break polymers down into monomers, and these monomers can be recycled in the cell. For example, if it is a protein, it can be cut into smaller pieces of amino acids that can be used to build other proteins by the cell. These enzymes which are present in the lysosome, as I've mentioned, break down larger molecules. If they were to be released in the cytoplasm, that will be very catastrophic because of the nature of these enzymes. That is why all lysosomal enzymes are always injected in the lumen of ER when they start their synthesis. So there is no chance that these enzymes will ever see cytoplasm. These enzymes not only degrade proteins that are cellular in origin, they also degrade structures, proteins, macromolecules that cells ingest through a process called phagocytosis. Cells also need to eat. When they come across a food particle they are interested in having or eating, they engulf it and enclose it in a vesicle. This vesicle, which is called a phagosome, when it is present in the cytoplasm, a vesicle containing the lysosomal enzymes, at this stage it is called the primary lysosome. It fuses with the phagosome and results in secondary lysosome. The lysosomal enzymes will go to work. They will start degrading the large molecules that have been ingested into smaller components and be put into the cytoplasm where they can be recycled. Any undigested material is expelled out of the cell. Also, I would like to mention that not only phagosomes fuse with the lysosomal vesicles, the organelles which have become non-functional in the cell, for example mitochondria, when they become non-functional, cell puts a membrane around these mitochondria, and these vesicles, which now contain cellular mitochondria, are fused with the lysosomal compartment. Mitochondria is degraded, and the components which are making the mitochondria are released into the cytoplasm for recycling purposes. So basically, lysosomes are very important in degrading material and providing cell the raw material for recycling.